My name is Jing Chen. I'm a CEO and founder of HiCom. I founded this company 10 years ago. We are doing microfluidic contract manufacturing. Before that, I worked as a professor in Peking University. And before that, I worked in the area of MEMS. I worked with Professor Preclinical in the University of Michigan. So today, I'm going to give you a brief introduction of microfluidics. What is microfluidics and how it works? And then I'll give you some examples of applications of microfluidics in preclinical research. So what is microfluidics? There's a short term of definition of microfluidics. Actually, microfluidics is everywhere. The capillary network within our body and within a plant, they're all at this level, at micron or nano level. So this is the nature of life. So microfluidics is science and technology that deal with a small amount of liquid. So typically we have two metrics to measure to decide what is microfluidics. One is that if one dimension or one critical dimension of this device is less than 500 microns, we call it microfluidics. Or if we are handling a liquid with the value of nanoliter or picometer or less, we call it microfluidics. But this definition is kind of too technical. It's hard to understand for most of the guys if you have never heard of microfluidics before. So here is the better one. We also call microfluidics lab on a chip, because everybody is familiar with labs. Here in Aquarius, we already have a lot of very good labs doing different life science research. So if you work with a working lab, you probably deal with these tubes or discs or flags manually. But then I think that I will skip this one because I think our next speaker is expert of lab automation. Then you go further to using this robotics or liquid handling station to handle all these procedures, all these experiments in an automatic way. And also I think these robotic or liquid handling machines can integrate all the function together. So what's the microfluidics? Microfluidics the idea is to bring this manipulation of liquid, not manually, not with robotics, but within a tiny little chip. We call it microfluidic chip. So in this way, it can do it automatically and integrate different functions. And then the unique advantage of microfluidics is the miniaturization, because we can incorporate all these functions of liquid handling into these tiny little chips. So the reader can be very small, and theoretically, we can build the whole lab on this tiny little chip. But there's always a but. In reality, this microfluidic chip, like this one, it has to work with a reader or an analyzer. So the idea is these tiny little chips work as a consumable or cartridge. We'll put the cartridge into the instrument and then get a result. So in reality, it has to work with an instrument. But at this talk, let us concentrate on this part, microfluidic chip itself. So there is a lot of applications of microfluidics. I think the top three is point of care, POC, and the next is clinical diagnostic, and the other is life science tools. I think this forum is more focused on life science tools for pharmaceutical research. So I will give you a brief history of microfluidics. So microfluidics, as I have talked with Eric, the first generation of microfluidic product is actually used in the International Space Station, ISS. They use that tiny analyzer to monitor the health conditions of astronauts on the ISS. I think this date back to 1995. And then this technology came back to Earth. And then there's a lot of companies working on this. They use this idea to build, a lot of good point of care devices to do chemistry analysis, as well as immunoassay analysis. This is the first wave of microfluidics. But then there's an empty period. During that period of time, there's no breakthrough or innovations. But then after the year 2015, there's a new wave of technology breakthrough, which focused on molecular diagnostic, including gene sequencing. So what happened during this period of time? There's a company, I don't know if you're familiar with the name, Theranos. It's founded by a girl named Elizabeth Holmes. There's a TV, there's a lot of reports on this because historical company that declared they can do over 200 types of analysis within maybe half an hour, but at that time it's not practical. When I read the report, I think, how can she do that? But actually he raised maybe billions of money, then his company, actually was fake. So I know there's saying in Silicon Valley, first you fake it, then you make it. But the problem is that she never make it. You fake it and fake it again. But technology keep moving even during this period of time. Right now, I think for advanced microfluidic devices like Cepheid, actually they can handle thousands, over 2,000 different analyses within this tiny little machine, and within 1 hour. That's very close to the idea of Theranos. So then there is a microfluidics 3.0, I think, in the future. So what happened next? Because I don't know if you heard about or working on a single cell, single molecular analysis. The idea is that right when we are doing something like PCR, like gene sequencing, we are getting result of average cells or organs. But this is good. But in the future, I think we probably want to see more precise pictures of the whole genome, of the whole cell, to identify, particularly identify which cell, which DNA has mutation, and which will help us to identify how the disease are forming and how to develop a certain kind of therapy. So right now we can only see one dimension. One dimension is how is the how much copies or virus in a PCR test. But in the future, we want to see more. We actually want to get 2-dimensional, 3-dimensional pictures of each cell or each molecular. So this so microfluidics gonna help us to realize this. So what happened next? Because even we can see a more precise, more clear picture of the biology, of the issues of the cells. Then what happened next is first we do diagnosis, then we move up to the treatment. So in order to facilitate the drug development, is one of them. We have to be able to do something that beyond reading the biology. We probably want to editing the genes, right? I think year 2022 chemist Nobel Prize was awarded to professor in Berkeley. They are doing gene editing, right? Then storing, then organizing, or even synthesis of cells and moleculars or biomarkers. This will definitely lead microfluidics. Why? Because when you take a picture of this, you can see that cells at the level of microns, 1 micron to 10 micron, and then the genes and also virus, and they are all within this range. So definitely you need some technology to identify them, separate them, and to make to collect all these things and to make it happen. So why really microfluidics? Because we are dealing with our object within these dimensions. So then I will give you a brief introduction of what material or how to make a microfluidic chip. This is the thing that I'm working on. So in microfluidics, there are three main materials used in microfluidics. The first one is polymer. The second one is silicon, and the other is glass. So polymer. So we are there are two kinds of polymers. The one is elastic. One is PDMS, and the other is thermoplastic. So PDMS is unique. I think maybe some of you are familiar with PDMS. So PDMS is kind of golden standard for prototyping a microfluidic device. The PDMS was first introduced in this industry by George Whiteside. He's a professor in Harvard and also pioneer of microfluidics. I think in the year 1990, I introduced PDMs. Then PDMs became a golden standard when you do, microfluidic prototyping. Then plastics. So today, most of cartridges are made of plastic because there's a lot of selections. There are different types of plastic, which means you have more choice. And then the plastic, most of the plastic made by injection molding, and which is cheap, cost efficient, because microfluidics are regarded as a one-time consumable. So cost efficiency is crucial in order to commercialize this device. So then the silicon part, which is the best part of microfluidics, because it's capable to produce feature size as small as microns, even nanometers, and other nanometers. Today, some advanced microfluidic applications like third generation or fourth generation NGAs are using silicon as their the most important one of the most important components. Then a glass for gene sequence, which is very useful in gene sequencing, or in some unique applications like EWOD, electrowetting. And there are other materials, but it's their application is limited. So the fabrication technology for polymer-based device, the biggest, the dominant technology is injection molding. Then hot embossing, because the hot embossing, the cost will raise, I think, by at least an order. So it's not used that it's not widely used as injection molding. Then soft lithography, which is used for manufacture PDMS. And then there's another routine of manufacturing which follows the technology of semiconductor technology. So the idea is first you use a mask, then you do photolithography, then you do something, some action of set blasting to form these tiny little structures. Then there's back-end process. I would say that the back-end process probably will make up as much as 80% of the cost of the total chip. So then there's a quantity control. So there's a lot of things to do. I cannot cover it in this talk, but I would mention that you can look into among the supplier. This is the map of global supply chain of microfluidic manufacturers. Actually, we are here. We are really comprehensive. We can handle silicon, glass, and polymers in-house at our factory. And then I will move to the microfluidic applications in preclinical research, I think you guys are familiar with this drug discovery procedure. So preclinical research is here, and there's a lot of R&D effort happens in other process. So I'll be talking the seven different applications because time is limit. So I will just give you a brief introduction. You can take the slide. Yeah, I can send this slide to you. You will want. You can get it from Eric and from the organizers. The first is dropping the digital PCR. This is the droplet from bell rate. So they primarily use for oncology applications because they are actual high sensitivities. And so what inside is something like that. So this is a droplet generator, and there are some tiny little structures at the bottom of the cartridge, which are used to formulate a tiny little droplet with a diameter of tens of microns. Another example is single cell analysis, which is very useful to explore cellular diversity. So this is the I think the idea is similar. They incorporate one DNA segment or one molecular within this within each droplet. So in this way, they can actually measure each droplet to determine the performance of the DNA incorporated inside, trapped inside the droplet. So in this way, they can get a map of all the cells within one test. Another one is liquid nanoparticle, LNP technology. This technology is famous. Everyone knows mRNA vaccine. So in order to generate to deliver the mRNA vaccine, you have to do this nanoparticles. So what's inside? What it looks like? So inside this nanoparticle machines, there is a little cartridge which do a mixture of different materials and generate nanoparticles here. In this area, there are very tiny little. It's a multi-step channels that used to generate nanoparticles here. Now you cannot see here, but actually this is very unique and good design. And then there is a 3D cell culture and organ chip, which is a very hot topic here. So I list, two of the leading companies in US and China. Actually, we are supplier of this company. They use different materials and different approach to actually the idea is to culture to mimic the living cells and organs, the cultured organs on either on a chip or on a microplate. But yeah, certainly with this kind of modifications, you can see that at the bottom of the on the microplate, there are reservoirs, there are channels used to deliver the nutrition to culture the cell. And then there's cell sorting and analysis. This is a company located in San Diego. So they are doing cell sorting, which is very useful for enrichment of real cells like CTC, right? So they use a piezo electric actuator control on the direction of the flow to separate different cells. And we are a supplier of one of the crucial part of this microfluidic cartridge. This is made from PDMS because of the accuracy, and also I think because of accuracy, you can only use PDMS in this scenario. We cannot. It's hard to manufacture thermoplastic part at the same accuracy and dimension. Then there's point of care. I just list a bunch of devices here. This is Apsis. Why they have this satellite here? Because they are, and they have, you know, they have a logo of NASA here because they actually the first generation of that device actually used in the International Space Station. So this is a space technology, but ultimately we by the effort of a bunch of engineering, this technology finally get back to Earth. So then there's liquid biopsy, which is used for non-invasive diagnostic of cancers. And also here are some two examples of chips, and these chips was developed by a professor in the University of Michigan and commercialized by a company also in California. And they have a huge amount of PDMS chips, 5K per month. So this we also a supplier of this chip. So in sum up, microfluidics has a transformative impact of life science, and in terms of versatility, and also it's very useful for future, you know, precision medicine or personalized medicine. And in the future, microfluidics will open up a new vision, not only able to read the bioinformation precisely in a three-dimensional way, but also can handle it, can read, edit, and synthesis all these cells and moleculars, which is very helpful. And one last word before diving into your next microfluidic project, you can reach out to an expert. It's I think it's probably more easier than ask, you know, crystal ball. So thank you. Here, this is my linking, and you can download some of the material by scanning this QR code. Yeah, there's a lot of, you know, microfluidic POC diagnostic product on the market. Sci-Fi, I think they achieved 4 billion revenues during the COVID, but even right now, I think they still have revenue of 1.5 billion per year. And also a film array, which is used for molecular diagnostic, and also Apsis for chemistry analyzer. There's a lot of companies working on this. I think for the diagnostic market and total market, global market is something like 10 billion.
Other questions? I have one question.
Okay.
What do you think the most exciting device or sensor is going to be released in the next year or two? Of all you could pick, what's the most exciting?
Definitely NGS, next generation gene sequencing. Yeah. When we see from 15 years point of view, I think NGS will replace PCR or molecular diagnostic eventually.
Very good. Thank you very much.
Okay. Thank you, Eric.